Welcome to the August edition of the Distinguished Lecture Series in the Trivedi School of Biosciences at Ashoka University. Our speaker today is Professor Partha Majumder. Partha is the National Science Chair, Government of India, and the founding director, and now a distinguished professor at the National Institute for Biomedical Genomics, which is a DBT institute. He is also currently the president of the Indian Academy of Sciences, which is one of the three national science academies. Professor Majumder obtained his PhD from the Indian Statistical Institute in Kolkata. And thereafter, he did work on demographic and population genetics at the University of Texas Health Science Center in Houston. In 1983, he started as a faculty member at the Indian Statistical Institute and was also a visiting professor at the University of Pittsburgh for a couple of years. Partha's research contributes to applying statistical, molecular genetics, and anthropological methods to various issues of human genetics, evolution, and disease. He's very distinguished in the field, and we are extremely pleased to host him today as our distinguished speaker. So, Partha, it's over to you.
Thank you very much, Professor Jamil. This is really an honor to be called upon to deliver this talk. Am I audible? Is my voice clear and sound okay?
Yes.
All right. So, again, like I said, that I'm actually very pleased and honored to be giving this talk. Let me share my screen with you.
Yes, we can see.
Good. So I have titled my talk as In Search of the Drivers, and Shahid has actually annotated my title by saying that it is in search of the cancer drivers. Indeed, we are looking for the cancer drivers, and I will define what I mean by a driver shortly. So I'm going to start with a statement that requires some amplification because it's not quite obvious to most people, even in the field. So the first statement that I will make is cancer is a disease of the genome. Now, when you talk about a disease of the genome, one thinks that it will run in families, it will run from one generation to another, because that's what we mean by a genetic disease. Cancer is a disease of the genome. It's a genetic disease, and therefore it better be heritable. So it should pass, it should move from one generation to another. The disease should aggregate in families, etc. But all of those standard hallmarks of a genetic disease are betrayed by cancer. Cancer does not follow those standard hallmarks of a genetic disease. Why then do we call cancer a disease of the genome? I will, as I go along, I will tell you that cancer is a disease of the genome in the following sense, that there are genomic changes, there are changes in the DNA of an individual that leads to cancer, but these genomic changes are confined to that individual and will not be passed on from one generation to another, meaning that these are in the somatic cells of the individual that do not participate in the process of reproduction and therefore are not heritable. So predominantly cancer is a disease of the genome because there are genetic changes, but these genetic changes are not heritable, and that's what you know. That's the amplification that I needed to provide. So if you talk about a genetic disease, mostly it will be familial, like I said, but in cancers, it's only about 10% familial, 90% of cancers are non-familial, yet we call cancer as a disease of the genome, primarily for the simple reason that, you know, the DNA changes take place, but these are in the somatic cells that are not passed on. When we talk about a cancer genome, the genome that actually participates in the process of cancer, It usually involves multiple steps, multiple mutations, and these are, like I again emphasize, that these are predominantly in cells that do not participate in the process of reproduction. The cells that participate in the process of reproduction are called germ cells, and those mutations are called germline mutations. Only about 10% of cancers are because of germline mutations. The remaining 90% are because of somatic mutations. So I'm going to define a driver, like I said, that the word driver requires a definition. Right now, I'm only going to conceptually define it, and at a later point of time permitting, I will define it more quantitatively and more statistically. So imagine, if you will, that these are a set of cells that are very neatly stacked, and imagine that that line below which it's stacked is the epithelial layer. So these are just cells below the epithelial layer. They all look the same. At birth, they all look the same. And as we move through our life course, these cells, the DNA in these cells will start accumulating changes. These are called mutations. And so they will start accumulating mutations. So what we're going to do is sort of simulate the life course, and as this individual gains age, these mutations take place in these cells. There are two different kinds of mutations that have taken place in these three cells, denoted as white and yellow. They are not exactly the same mutation. Two of the cells have the same mutation. The third cell has a different kind of mutations, but these mutations are essentially innocuous. We pay no attention to it because they just happen in the background, and we don't really need to bother about them. And this continues. So here are some more mutations that have arisen. Some in cells that where there were pre-existing mutations, and some in newer cells. Again, like I said, that most of these mutations will be innocuous. We don't pay any attention to them. But sometimes mutations need to be, one needs to pay attention to these mutations, because sometimes what these mutations do is that they make photocopies of the cell in which they have arisen. So these are like clones. This is clonal propagation. This is not how cells normally divide. Cells normally divide from 1 to 2 to 4 to etc., like a 2 to the power of n kind of a division. But here, what one sees is that from one cell, you suddenly find a proliferation, a number of copies of that same cell being made, and those are called clones. Again, these clones arise in our bodies all the time. We are completely oblivious to them because they cause no harm to the body. So some of these mutations are different from some others in that they produce clones. Again, here is a black mutation that arose in the background of a yellow mutation, and that gave rise to a small clone. But again, we don't really care about them. These are arising all the time in our bodies. We don't pay attention to them because we don't even get to know them. So this process goes on. So here is another mutation, the red mutation that has taken place in a cell that already had the black and the yellow mutation. But this particular mutation is different from the previous mutations in the following sense, that this particular cell gave this particular mutation gave the cell a huge growth advantage, and a large clone has formed. It has breached the epithelial layer and it has grown into a malignant tumor. So this particular mutation, the red mutation, is the one that drove this particular cell to becoming a malignant tumor. And we need to identify these red mutations because they are the culprits that are causing malignancy in an individual. So those are called the driver mutations, and that's what we shall be after. It's not easy to catch hold of driver mutations, primarily because they are happening in the background of all of the pre-existing mutations and some mutations that will occur later as well. So at a point of time when we are analyzing a tumor, we see a large plethora of mutations. And from that plethora of mutations, we need to identify these red mutations that are really the culprits. This is really a statistical enterprise. Well, it's a genomic enterprise because we need to sequence the DNA, or we need to understand the DNA where these changes have taken place. And then we need to analyze that sequence data, the genomic data statistically, in order to be able to identify the signal, which is the red mutation, from the noise, which is the background of all of the other mutations about which we did not care and we do not care. So that's what this enterprise is all about. And even after this red mutation has taken place, there will be other mutations that take place, and these mutations will arise. So the question that we are asking is, what are the genes and genomic alterations that drives a cell to becoming a malignant tumor? These driver mutations give the cell a huge growth advantage. And as a result of this huge growth advantage, the clonal propagation, it becomes that particular cell attains the property of clonality and it becomes a malignant tumor. So that's what we shall be after. Now, the hypothesis that cancer may be a disease of the genome has been asked many years ago, maybe 50 years ago, maybe 60 years ago. Why weren't we able to address the question and get to a solution maybe 50 years ago? The reason being that the question was there in the minds of our predecessors, but they did not have the technology to identify at the genomic scale, and they did not have the technologies. And many people also did not believe that that's the way to go. And so what I'm going to do is to justify the technology that we currently use in order to identify these tumors. This particular statement that I'm going to read out, a very short passage from a paper that was published in Science in 1986 by Nobel laureate Renato Dulbeck. The title of his paper was "A Turning Point in Cancer Research: Sequencing the Human Genome." This was pre-Human Genome Project days, and there were different kinds of justifications that were being given in order to embark or in order to undertake the Human Genome Project that entailed the sequencing of the entire genome. And people like Renato actually were big proponents of sequencing the entire genome. This is what he said: "If we wish to learn more about cancer, we must concentrate on the cellular genome. We have two options: either to try to discover the genes important in malignancy by a piecemeal approach, which is what our predecessors did, by the way. They looked at specific portions of the genome where technologies existed, and they identified the changes in that and tried to ask themselves whether that particular mutation was associated with cancer in any meaningful way." So that was a piecemeal approach of looking at small portions of the genome. But what he said is that either we try to discover the genes important in malignancy by a piecemeal approach or to sequence the whole genome. And he said it will be far more useful to begin by sequencing the cellular genome. So not only the Human Genome Project had other kinds of justifications, but this was the justification for undertaking the Human Genome Project, or even to sequence the whole genome for understanding cancers in a holistic way. And subsequent to that, all of you know that the Human Genome Project was undertaken. The first human genome was sequenced. That led to a proliferation of certain kinds of technologies. But more importantly, in the last 10 or 15 years, it has spawned a huge technological breakthrough, as a result of which we are able to sequence cellular genomes at scale and at speed. And I'll give you a comparison. So, for example, when the first human genome was sequenced, whatever technologies that existed, the best of technologies were used, and to sequence one single genome, it took 13 years. Today, in the peri-urban center where I conduct my research, it's a place about 50 kilometers from Calcutta, in a peri-urban region called Kalyani. In our institute, right now, we can sequence like 4 genomes a week. So that's the power of these technologies. 13 years for one genome to 4 genomes a week. This is what we can do. And we are not in the United States. We don't have the best of technologies accessible to us, or multiple copies of the same technology available to us. So that's the power of this technology. And because of this ability to do so, we are able to answer some questions. And I'm going to share with you the questions that we have been able to answer using this particular approach, using these technologies. But before I come to the main part of my talk, let me start with a couple of stories. The first story is from, both of these stories are real, true stories. The first story is from the pages of the oldest medical journal in the world, called New England Journal of Medicine. So this is, I'm just going to show you pictures and paraphrase that story for you. So this is a picture that was published in the New England Journal of Medicine. This, as you can see, it's a whole body image of an individual, and these black dots are cancers. So we don't even know where this cancer arose in the body because it's completely spread throughout the body. It has metastasized throughout the body. And this is the picture that this individual came when he presented himself to the doctor. The doctor then decided to treat this patient, and he used a specific kind of treatment and used that for a period of time. For about 2 weeks at a specific dosage. And after that, again, a whole body scan was done. And this is what happened to that individual. Gone, completely evaporated. The tumors don't even exist in the body. So this individual was very lucky. He responded to the disease, and this is what happened to the individual. The same doctor, after a period of time, there came another individual to the same doctor with nearly the same body image. And this, the doctor then treated that individual also with the same drug for the same period of time at the same dosage, and then took a whole body scan. And this is what happened to the other individual. Did not at all respond. So the tumors remained where they were. And so the question that we ask is, why is there a smiley in that particular arrow and a sad face for this individual? So this is a story that we'll come back to, where, and I'll tell you the reason for this, and those particular kinds of solutions or understanding arose as a result of, again, genomic sequencing and genomic understanding. Story number 2 is a personal encounter. And I'll tell you the context of it. This involves a teacher of mine. Like Shahid said, that I went to the Indian Statistical Institute, where statistics is what we primarily learned, but we also had to learn other kinds of sciences. So this was a teacher of mine who taught us geology. He was a heavy smoker throughout his life, and we could see him smoke on the campus. At that time, smoking was quite rampant and quite popular. So he was a big time smoker. He retired, and I knew the family. I actually knew the family quite well. So one morning I get a call from his wife saying that, he's not doing well. Her husband is not doing well, and the local clinician said that he has cancer, probably has lung cancer. So since I was working on cancer, she asked me if I could put her in touch with the doctor. And of course, I did put her in touch with the doctor. They were also not very well to do. So we went to a doctor who was, practicing or who ran a cancer hospital, quite in a semi-urban area. And anyway, that was quite far from her home. So this is one morning where she and the doctor are walking on the corridor of the hospital, and I'm sort of tiptoeing them from behind. And that's a conversation that I'm overhearing. So I reproduce the conversation. The doctor says, well, the patient has NSCLC. Don't worry about what NSCLC is. It's non-small cell lung cancer, but we'll just say the patient has lung cancer. I need to put him on chemotherapy. As soon as the wife hears the word chemotherapy, her tentacles go up. The reason being because she knows what chemotherapy means, that her husband is going to feel quite sick as a result of chemotherapy. She has to be on his bedside, and they live quite far away, so she has to move up and down from her home every morning and then go back in the evening, etc. So all of these thoughts come to her mind, and she asks a very natural question. She asks, can the chemotherapy be done at home? The doctor's reaction is immediate. He says no. Then he says, wait a minute, let's check out his EGFR mutation status. So here is something, the doctor is speaking genetics and is saying that an answer to that question, whether the chemotherapy can be done at home, will come depending on the result of mutations in the EGFR gene. So EGFR is a gene. It's called epidermal growth factor receptor. So he's going to find out whether that particular gene has mutations or not. And then he explains to the wife, if it turns out that he has EGFR mutations, then you can take him home and administer chemotherapy orally. So it's a double whammy in the sense that you can not only take him home, but you can actually administer chemotherapy orally instead of injectable chemotherapy. We'll come back, we'll return to both of these stories at the end of my talk. The results that we have gotten are because we participated in an international consortium called the International Cancer Genome Consortium. India participated, and in India, we represented genomics and the Tata Memorial Hospital Advanced Center for Cancer Advanced Center for ACTREC, Advanced Center for Treatment, Research and Education in Cancer. That's a part of the Tata Memorial Research arm of the Tata Memorial Hospital that participated as a clinical arm of this particular national project that we undertook. The goal of the International Cancer Genome Consortium was to obtain a comprehensive description of genomic, transcriptomic, and epigenomic changes in 50 different tumor types or subtypes that are of clinical and societal importance across the globe. So that was the express goal of the International Cancer Genome Consortium. It was done in a cooperative, collaborative manner, and India participated with oral cancer as the mainstay. So we were going to look at oral cancer, and that's we have obtained results pertaining to oral cancer, and those are the results that I'm going to describe to you. Like I said, that the project team comprised ACTREC, the clinical arm that was led by Dr. Rajiv Sareen, who is a clinician, and the genomic arm was led by the National Institute of Biomedical Genomics with Dr. Maitra, Arindam Maitra as the leader in the wet lab, and Nidhan Biswas as the leader in the computational lab. It was funded by the Department of Biotechnology, and I coordinated this entire project. Cancer of the oral cavity is the eighth most most common cancer in the world. About 260,000 new cases arise annually. About 2/3 of them arise in developing countries. About 130,000 deaths happen annually, and they account for about 1/3 of all tobacco-related cancers in India. And it's also associated with human papilloma virus. Why are we, okay, so I also need to tell you that when we look at the oral cavity, or at least in India, we are looking at one particular region of the oral cavity, which is really the oral cavity excluding the tongue. The oral cavity excluding the tongue is called the gingivobuccal region of the oral cavity. And why did we exclude the tongue? The reason why we excluded the tongue is because of epidemiological data that we already had when we started our project. And that was a stark contrast between India and the Western world. In India, about 25% of the oral cancers of the oral cavity are because of tongue cancers, and about 75% are in the gingivobuccal complex, gingivobuccal region. The contrast is with the West. About 25% of cancers in the West are in the gingivobuccal region, and 75% are tongue cancers. So in the West, there was already a project that was initiated called the Cancer Genome Atlas project that was anchored in the US, and they were also researching on cancer of the oral cavity. When we started our project, we knew that most of the patients in the US project would be tongue cancer patients. And so what we decided is that let's generate data on the gingivobuccal complex, and at some point of time, then we can compare our results with the US results and see what are the major differences in drivers between gingivobuccal oral cancer and tongue cancer. So that was the reason why we restricted ourselves to gingivobuccal oral cancer, excluding the tongue of the oral cavity. The combination, as all of us know, is essentially the culprit is tobacco, also areca nut, which is a psychoactive substance. Very, you really get habituated to areca nut, and you can't really leave. So this is very important, that tobacco in combination with areca nut. Also, it's a sleek lime is used. The lime actually chews up the epithelial layer, and the carcinogens in tobacco that then go and hit the cells directly. So that's really the hazardous combination. Areca nut is the fourth most common psychoactive substance in the world after caffeine, alcohol, and nicotine, and its use extends to several hundred million people, primarily in South Asia, including Southeast Asia. So right now, what the data that we have collected and analyzed is about 400 patients. We need to go up to 500. We still have another 100 to go, but this is an interim result based on 250 patients. Most of these patients are male. 85% are males and 15% are females. This is the most common cancer among males in India. Most patients are young. The median age is about 48. Most patients are tobacco users. As a matter of fact, almost all of the patients are tobacco users. We did not find any significant human papillomavirus involvement in gingival oral cancer. We carried out resequencing of the genome, and we did resequencing of paired blood and tumor DNA. The reason why we sequence blood, which is really not of importance to us or not of interest to us, we are more interested in the tumor DNA, but we also wanted to weed out all of the germline mutations because we were looking at somatic drivers, and the germline mutations would be present in the blood. They would not have somatic mutations. On the other hand, the tumor DNA would have a combination, or would have the totality of the germline mutations and the somatic mutations that have accumulated during the life course of that individual. So imagine, if we will, that if we subtract out the mutations that we find in the blood from the total number of mutations that we find in the tumor, we would be left with somatic mutations, and then we could analyze that data in order to identify drivers. We used two orthogonal sequencing platform, at least initially, because we wanted to be absolutely certain that we are generating high quality data. This is early days. This was, I'm talking about 2009-10, when we were using orthogonal sequencing platform in order to make sure that we are generating high quality data. And then we also validate the data using other kinds of sequencing platforms. We also use DNA microarray for copy number, identifying copy number variations, large structural variations, and then we moved over from exome resequencing to whole genome resequencing as time went by, and these platforms became more and more robust. The depth of coverage, we use about 35 to 40x, both in the blood and in the tumor. And we generate high quality data using orthogonal platforms. We have in excess of 90% concordance of various kinds of DNA variants, and between DNA microarray and sequencer in excess of 99%. So essentially what we are saying is that the data that we are generating and analyzing are fairly high quality data. Want to make the first statement that I wish to make is with respect to the mutation rate. With some patients, as you can see in the top left-hand panel, the vertical bars are the total number of mutations. As you can see, most of these patients would have smaller number of mutations, but there are some individuals who have a very high number of mutations, as you can see in that histogram. The mutation rate per MB is about 3. So you get about 3. The median number of somatic mutations is about 3. This is in gingivobuccal oral cancer. If you look at the TCGA data, the Cancer Genome Atlas data, you find that in other parts of the head and neck, in other parts of the oral cavity and other parts of head and neck, the mutation rate is higher. It's 5 compared to 3 per MB. The number of somatic variants that you find in the somatic variant and in small insertion deletions, the median number per patient in gingivobuccal is about 86, and the median number in other parts of the head and neck squamous cell carcinoma is about 140. So both in terms of mutation rate as also in terms of the total number of somatic variants in the head and neck cancers, the gingivobuccal region has much smaller reduced mutation rate and reduced number of somatic variants. Excuse me. Most of these somatic mutations that we find are actually non-synonymous. There's a huge interpatient variation in mutational landscape. This is the on the x-axis are patients, and on the y-axis is the number of mutations. Number of mutations are silent, have been categorized as silent and non-silent. The yellow portion is the silent mutations, and the blue portion is non-silent mutations. As you can see that there is a huge inter-individual variation in the mutational landscape. When we looked at these mutations, one of the questions that we are asking is that, can we find the reason for these mutations? And what do we mean by can we find the reason for these mutations? Well, if you look at mutations and you look at specific somatic mutations, and if you look at the context in which these somatic mutations is arisen, genomic context, you can actually find that there are certain kinds of signatures that these mutations create with respect to the context. I'm not getting into the details of these signatures, but essentially these different kinds of signatures arise because of the way these mutagens or carcinogens act on the DNA. Again, that's biology. I'm not really getting into that in any great detail. But what the Wellcome Trust Sanger Institute had done is that they looked at a large number of cell lines from different contexts, and they identified 30 different mutational signatures. And these mutational signatures would have, and let me just show you a few. Let me just show you the mutational signatures that we find in gingivobuccal oral cancer. And so these are mutational signatures that one finds, and these have been associated with like tobacco, ultraviolet radiation, an endogenous enzyme called ApoBec, and so on. So as you can see from the left-hand side panel that there are spikes. X-axis are the various mutations, and the y-axis is the number of mutations, and there are regions in the DNA, there are contextual spikes in the DNA, and those can be associated with certain kinds of exposure to various kinds of carcinogens. So the upshot of our work in OSCCGB, gingivobuccal oral cancer, is essentially this, that tobacco leaves a telltale signature on the genome, and we find that telltale signature in gingivobuccal oral cancer. We also find other kinds of mutation signatures, ultraviolet radiation, ApoBec, the endogenous enzyme, and other kinds of mutational signatures. But these are, of those 30, these are the 4 predominant mutational signatures that we find in OSCCGB. We generated these data, we analyzed them statistically. I'm not getting into the statistical analysis of the data. And then we identified what are the genes that drive. Remember, we asked this question, what are the genes and genomic variants that drive these mutations? So these genomic variants were mapped onto the genes, and the genomic drivers were, variant drivers were mapped to the genes. And what we find is that there are 10 genes that drive, and that these bar diagrams give you the percentage of patients with different kinds of mutations. Again, without getting into the details of what these different kinds of mutations are, let me show you the genes that are involved. The one that takes the biggest hit is TP53. P53, of course, has been implicated in various kinds of cancers. In almost all cancers, P53 is involved. In your buccal oral cancer, that's a major driver. Mutations in P53 is a major driver. Followed by caspase-8, FAT1, NOTCH1, PIK3CA, HRAS, and so on. So there are these genes that are significantly mutated in order to drive the cell to becoming a malignant tumor. And most of the time, you will not get one particular gene that's involved. You get a combination of these 10 genes that are involved in any single cancer patient. There is another characteristic that one needs to emphasize. A gene such as P53 can work both as a tumor suppressor as also an oncogene. In the context of oral cancer, all of these genes act as tumor suppressors, which is really bad news, but I'll tell you the bad news in a minute. They act as tumor suppressors, and that was again a finding. We made this finding using cell line data as also by looking at the distribution of mutations in these genes. When you have an oncogene, usually there will be a hotspot. There'll be a region in the gene where most of these mutations will take place, and that's a characteristic of an oncogene. On the other hand, when you have tumor suppressors, the tumor suppressors are usually active all the time, as opposed to the oncogenes, which are mostly asleep. When a mutation takes place in a tumor suppressor, either the level of expression goes down or the gene goes to sleep, essentially. It doesn't produce any protein. So any mutation in the gene, will any non-synonymous mutation in the gene will probably have an impact and will make that active gene reduce its function or even make it go to sleep. So usually any place, a mutation if you look at mutations in a gene, and if it's distributed throughout the gene, and if it's a cancer gene, then you would tend to believe that it is a tumor suppressor. You start with that hypothesis, go back to cell lines derived from patients, and then find out whether it's really working as a tumor suppressor, or you can do RNA sequencing to find out whether it suppresses the level of the protein in the context of these mutations. And we've done both. So essentially, these are tumor suppressors. So the upshot of all of this is that oral cancer is predominantly driven by mutations in genes that act as tumor suppressors. I talked about these single nucleotide variants that are single nucleotide mutations. You could also have other kinds of mutations like insertion deletions, copy number variations, and larger structural alterations. And again, these copy number variations, larger structural alterations, etc., they will bring in more genes. And there are a small number of genes in which structural alterations are important, etc. But predominantly, these are driven by single nucleotide mutations, but other kinds of changes, DNA changes also matter, and they evoke changes in other kinds of genes in the context of oral cancer. Given this knowledge, how can we probably use this knowledge in identifying or making use of this knowledge? One way to make use of this knowledge in genetics, it's mostly prediction. You want better management, prediction leading to better management of the disease. So in this particular case, when we look at these single nucleotide variants and look at these patients and try and group these patients by the nature of their mutations, what we find is that P53 forms a cluster of patients. Mutations in P53, forms a cluster of patients, and similarly, mutations in FAT1 and or caspase 8 forms another cluster of patients. The third cluster is a set of heterogeneous clusters. So what we look at is that even within these larger clusters, there are smaller subclusters. Without getting into the details, those subclusters are subclustered because they have mutations in other kinds of genes. These gray bars that you see at the bottom are survival, months of survival after treatment. And what one could do is, depending on, and as you can see that these bars are not all of the same height. There are some bars that are significantly different from other bars, meaning that survival probabilities are quite different for individuals having different sets of mutations. And that's the kind of prediction that one can make. Knowing the kind of mutations that this particular patient has, you can probably make a statement unless you manage the disease fairly well, meaning that ask the patient to come back, the disease will recur and the patient will not survive. But if you ask the patient to come back repeatedly, you can probably manage the disease better. So these genomic data have predictive abilities, and they enhance the life of the patient after treatment, even though as of now, we can't use these data in order to block the disease or provide direct treatment to the individual. But again, I'll shortly come to a treatment angle of this. So again, we've done some cell line experiments to show what happens if you knock down FAT1 and caspase 8 in cell lines, etc. And we have actually shown that they lead to increased cell migration that's involved with metastasis and also clonal growth. So anyway, we published this paper. We then wanted to find out, like I said in the beginning, that we are looking at cancer of the oral cavity without the tongue, and West is doing the tongue, so we wanted to compare. So essentially, when TCGA published their data, what we found was that they identified 11 driver genes. So like we identified 10 driver genes, they identified 11 driver genes. So the obvious question was, are we identifying more or less the same set of genes? The answer is yes, by and large, but there are important variations in the proportion of patients who have mutations in that particular gene. So if you look at, for example, caspase 8, which is the second row, the first column is the name of the gene. The second column is the proportion of patients that are associated with mutations in that specific gene. And the third column is the proportion of patients associated with that gene in the TCGA data in tongue cancer. So if you look at caspase 8, for example, you see that 43% of our patients in India, gingivobuccal patients, have mutations in caspase-8. On the other hand, only about 10% of patients of tongue cancer have mutations in caspase-8. Even though caspase-8 is involved both in gingivobuccal cancer as also in tongue cancer, there are enormous differences in mutations, or in other words, enormous differences in the importance of these genes, of these driver genes in the two different kinds of cancers. We've also found certain other genes that are specifically mutated in gingivobuccal oral cancer, such as HRAS, KMT28, and ARID2. And I'll come back to ARID2 at the end of my talk. So again, the conclusion is that there are similarities in terms of the cancer driver genes, but the importance of these drivers are quite different in gingivobuccal oral cancer versus tongue cancer. Also in gingivobuccal oral cancer and also in tongue cancer, there are some specific genes, specific drivers that are specific to that region of the oral cavity. I want to turn to these two stories that I began with, and I'm going to tell you a little bit about another gene called BDAF. This is a gene, and I'll tell you the function of the gene in a second. Mutations in BDAF are found in malignant melanoma and in other cancers. So 70% of malignant melanoma are driven by mutations in BDAF. 30% of thyroid cancer patients and 10% of colorectal cancer patients. So BDAF gene is involved in multiple cancer types, but predominantly in malignant melanoma. What does BDAF do? BDAF is essentially a gatekeeper gene. So it's a signaling molecule. It signals the trafficking of cargo from outside of the cell to inside of the nucleus. So essentially, there's a lot of cargo, biological cargo that comes from outside of the cell to inside of the cell that needs to be regulated. And the BDAF gene is essentially a regulator of that cargo. If it is not regulated, if BDAF goes haywire, it doesn't function well, then this cargo keeps coming, and that leads to improper function of the cell. So if you have a mutated BDAF, so this is involved in cell signaling, as I said, cargo signaling. So so that's what happens in a normal cell. In a cancer cell, the BDAF gene is mutated, and when you have a BDAF gene mutated, it doesn't actually perform its proper function of signaling, and therefore the cargo is not appropriately modulated, and that leads to a signal that tells the cell to grow and grow and grow, which is a hallmark of lack of apoptosis or hallmark of clonal growth. So essentially, mutation in BDAF leads to clonal growth of the cell. That's again a hallmark of cancer, or a prerequisite of a tumor. So what one can do is then to block the effect of this mutation. One can find a drug and see if you have a mutation in BDAF, you find a drug that will specifically block the impact of that mutated BDAF, and then the cell will not. Then the signaling will be such that cell will not grow and grow and grow. So a specific drug that acts against a mutated BDAF will actually enable controlling of the tumor, and that's what is important to know. And now there are essentially drugs that are available called vemurafenib, for example, that will act on this mutated BDAF and will be able to control the cell from growing. So this is exactly the story that I want to come back to. Both patients more or less came to the doctor with more or less the same body scan. The doctor provided the treatment. One of them, in one of them, the tumors regressed. In the other person, the tumors did not show any change at all. When you go back and do the mutation analysis of BDAF, what one finds is that this individual had mutated BDAF. What was the doctor applying? The doctor was actually applying vemurafenib, which will only act when there is mutation in BDAF because it's specific to the mutated BDAF protein. The first individual had mutation in BDAF, so responded very well to vemurafenib. And as you can see, that as a result of that response, the tumors have evaporated. The second individual did not have any BDAF mutation, and therefore did not respond to vemurafenib. So the take-home message is that, first of all, understanding the genomic basis of cancers helps us to manage the disease better. And sometimes we are able to identify drugs that will act against these specific mutations, and that will become very beneficial for treating cancer. So, and this is one wonderful example. Like I said, that treatment is a big problem, and there are mutations in a large number of genes that take place in any cancer patients or in a group of cancer patients. So essentially, recurrence frequency distribution in the driver genes has a long tail. And of course, the first spike, as you can imagine, so I'll tell you why we need to bother about this long tail in a second. But as you can imagine, that this the spike that you can see in the first vertical bar is of course P53, because that gets the greatest number of hits. But there are these other genes that have smaller number of hits in a group of patients. Why do we need to bother about this long tail? Like I said, P53 works as a tumor suppressor in the context of oral cancer, gingivobuccal oral cancer, and it's essentially not druggable because mutations take place anywhere. The gene goes to sleep, and if you have, how can you have drugs that against a large number of mutations? So it's even conceptually that's very difficult. But what we said is that supposing we look at this long tail and we map these onto biological pathways, can we modulate the pathways? That was the question that we asked. And so what we did is that we mapped these genes on pathways. Again, I'm not going to give you details of the entire exercise, but the upshot is that we were able to map most of these mutations in these genes. These genes belong to a particular biological pathway called the arachidonic acid metabolism pathway. The arachidonic metabolism pathway has certain key enzymes called LOX and COX. This is important because there are LOX and COX inhibitors that are available, just as you had an inhibitor of the mutated BDF. Similarly, there are LOX and COX inhibitors that are available. As a matter of fact, aspirin is an inhibitor of COX. So you have these COX and LOX are involved in various kinds of features, cellular features that are associated with cancers: proliferation, anti-apoptosis, lack of death, and angiogenesis and invasion and metastasis, where cells proliferate and go and infect, go and metastasize to other tissues. LOX and COX is involved in all these various kinds of. So what we did was we looked at cell lines, and we found that weaker expression leads to weaker expression of COX2, leads to a higher apoptosis. We also have selective inhibitors of COX2, and we use those in a dose and time-dependent manner, and we found that stronger inhibition leads to higher apoptosis. So this is constitutively expressed in HepG2 cells, and therefore we used HepG2 cells in order to identify these kinds of things. So we were pretty sure that this particular arachidonic acid metabolism pathway that has two key components, COX and LOX, are very useful. We then, because we are not an institution who can do clinical trials, we are now negotiating with organizations that can actually undertake clinical trials in order to find out whether our basic finding of inhibiting LOX and COX will work or not. In the meantime, what we did was that we tried to find out whether the overall survival probability with mutations in these pathways or without mutations is going to be different. And what we find is that individuals who have mutations in these pathways, where LOX and COX are kind of automatically inhibited, they survive longer than those individuals who do not have mutations in this pathway. So inhibition might actually work. And that's what our finding was. And this is without any treatment at all. Just having or not having mutation enhances the disease-free survival probability by about 17 months. So I want to come back to the second story that I said. If you recall, the second story is a conversation that's going on between the patient and the wife of the patient. Doctor says the patient has lung cancer, need to put him on chemotherapy. The wife says, can it be done at home? The doctor says, no, we need to check out his EGFR mutation status. And if he has mutations in the EGFR, you can take him home and administer chemotherapy orally. So oral chemotherapy is something that's becoming very popular for treatment of certain kinds of cancers. The epidermal growth factor receptor is overexpressed in cells of the lung and breast cancers. It's associated with various features of cancer, again a signaling molecule essentially, and it eventually leads to uncontrolled cell proliferation when the EGFR mutation, when it's mutated. So, and these mutations take place in a specific region of the gene or of the protein that's involved with both survival and proliferation. It's the domain where most of these mutations take place is called the tyrosine kinase domain, and that's responsible for activating anti-apoptotic pathways. Anti-apoptosis is an abnormal cellular function. The cells refuse to die. Apoptosis is the normal function where cells will be born and will die after a certain period of time. In cancer, cells refuse to die, and these genes like EGFR, mutations in genes like EGFR, will actually activate these do not die signaling, do not die signaling to the cells. There was a major study that was done. This is called the Lux-Lung-3 study, where they looked at a particular selective orally bioavailable blocker of this particular family of genes of the EGFR. And they found that this is orally bioavailable, like I said, afatinib. The response rate was 56% compared to the injectable chemotherapy, which is platinum-based, which is called cisplatin. It's only 23%. So first of all, it becomes orally bioavailable, so you can use oral chemotherapy, which is good news, and the response rate is also higher with respect to the oral chemotherapy as opposed to the injectable chemotherapy. The disease-free survival also in patients with EGFR mutation is higher when you provide the oral chemotherapy as opposed to the injectable chemotherapy. And it's not hugely different, but it's like 5 months of additional survival. I'm running out of time, and I'm going to skip many of these. We looked at other kinds of genomic features, which is a part of the ICGC goal, International Cancer Genome Consortium goal, which is to look at transcriptomes and also methylomes. And we have looked at methylation in the context, or the role of methylation in the context of expression, and we've been able to identify some important things. But again, I'm essentially going to end here just to tell you that when we looked at methylation and when we did an integrative analysis of methylation data, this is genome-wide methylation data, promoter methylation, of course, not gene body as of now. We've also done gene body methylation and its impact, but this is promoter methylation. And when we look at promoter methylation and its impact on expression, we find that the methylation actually controls certain pathways or impacts on certain pathways. I shouldn't say control, impacts on certain pathways, and like PPAR signaling pathway. Interestingly enough, the arachidonic acid metabolism pathway that we just discussed in the context of mutations also comes up in the context of methylation-driven dysregulation of transcription. And so we find that 5 different biological pathways are impacted on by methylation that dysregulates the expression of these genes, and these genes fall on 5 important pathways, and all of these pathways are actually associated or have been associated in one way or another with different forms of cancer. We've also looked at gene body methylation and have identified the central carbon metabolism pathway as being important in oral cancer. So, impacted on by methylation, the transcriptome data are actually involve 6 different pathways, including the oral cancer or central carbon metabolism pathway in oral cancer. We've also done a pan-cancer analysis of whole genomes, pooling data from ICGA, TCG, other ICGC, etc., and as a consortium, we've published a pan-cancer analysis of whole genomes. Don't have the data to, don't have the time to describe the pan-cancer analysis data, so I've only described our own data, which pertains to oral cancer. I'm going to skip all of this. We've also looked at metastasis, cell migration, and what leads to metastasis, but I really don't have the time to tell you that now. I do want to tell you what we are doing right now, just in 1 minute. I told you that there are 3 genes that are specific to gingivobuccal oral cancer, not involved with tongue. And one of those genes is called ARID2. And that caught our attention, and for various reasons. And we are trying to research on that. Cancer tumors are quite heterogeneous, both in terms of expressions of genes, in terms of both cell types and cell states. Cell states change, and cell types are quite different. So there's huge amount of cellular heterogeneity in cancers, and we weren't able to study this because we were looking at bulk transcriptomes. Very recently, we have the world has the ability to be able to look at transcription in single cells, genome-wide transcription in single cells. So we are able to generate single cell transcriptomic data, and we are able to find out the extent of heterogeneity in these gingivobuccal oral cancers. And as a matter of fact, this graph just tells you, this picture just tells you the kind of heterogeneity that we see. These are various kinds of cells, multiple cell types, B cells, epithelial cells, etc. This is in one patient, and if you look at multiple patients, you will see that there is a lot of difference among patients in terms of the proportions of different kinds of cells, and that, of course, has a story to tell in terms of cancer and its progression. We are actually looking at ARID2, and if you look at ARID2, the bottom panel will tell you that there are a whole set of cells that express ARID2 and another whole set of cells that do not express ARID2. So essentially, ARID2 can be in some ways can be interrogated in a subset of cells in these oral cancers, and that's what we are after, because in many patients we find ARID2 is actually mutated, and there are changes in ARID2 both in terms of transcription as also in terms of genomic variation. So we have moved from looking at tumors and bulk cells in the tumor to single cells in the tumor to address heterogeneity and to address which particular cells, whether these 10 different driver genes. Not every patient will have mutations in all of the driver genes, but let's say that they have mutations in 3 of the driver genes. Which specific subsets of the cells will have mutations in which driver gene is the question that we are asking. So this is early days in terms of our single cell work, but we hope to be able to draw some useful conclusions based on our single cell work. So the whole goal of our work on genomics in cancer is to be able to unleash the power of genomics to effect precision medicine and turn cancer into a chronic disease. Essentially, many of us, the elderly people, have, let's say, most of us or many of us will have hypertension. Essentially, what we do is pop a pill in the mouth in the morning and go about doing our daily chores and keep our blood pressure in check. So we are dreaming of a day when a cancer patient will be able to pop a pill in the mouth, and cancer will become a chronic disease. They will go ahead and do their daily chores without having to be tied to the bed. So this is what the ultimate goal is, and we are sort of inching our way forward there. And I end my talk here, and I'm happy to take any questions. And I thank you for giving me this opportunity, and I thank you for your attention. Thank you very much.
Thank you very much, Partha. That was a lot of work. Okay, so the way we do it is, if you want to ask a question, please raise your hand, unmute yourself. So Shashi, your hand is raised. Go ahead.
Yeah. So it was nice to see you, Partha.
Hi, Shashi.
good way, and a fantastic talk. You gave us such big, overwhelming, data-driven talk on on cancer drivers. So I just we can discuss offline many things. One major question I have is, because much of our understanding of cancer about cancer drivers versus tumor suppressors is from classical genetics. So now that you are doing whole genome analysis, there are all kinds of mutations occurring. Are there like a single gene, like for example, a kinase, activated kinase kind of a cases? Are there any cases where a same protein, two mutations leading to a driver kind of a case rather than a tumor suppressor?
You mean, does it work as an oncogene?
Hmm.
In the context of oral cancer, no, but in other kinds of cancers, oncogenes are indeed involved. Mm-hmm. So in the case of gingival oral cancer, we find that it's predominantly driven by tumor suppressors. And if you're asking me, are there any patients where the same gene that predominantly acts as a tumor suppressor in other patients, does it work as an oncogene in that patient? The answer is no. We have looked for that also, whether it's a mixture of both oncogenic action versus tumor suppressive action. Inevitably, it's all tumor suppressive action. We have not found oncogenic action.
Oh, I see. Okay. Yeah.
Okay. Kasturi? Shashi, you're done, right?
Yeah.
Hi, this is Kasturi. Sorry, my video is off. I have two questions, Shahid. Can I ask both now, or
yes, please go ahead.
So one is you as an expert, your opinion based on your findings, and the other is kind of technical. The first is, do you really think that the passenger mutations are absolutely inert, or they have certain level of gene interaction with the drivers that may change the makeup of the disease in a patient, or even their recurrence ability? And the technical question is, when you generate cell lines, do you really ensure that they are analyzed within a certain passage time, after which they may acquire further mutations that may not be present in the patient that they're derived from? Thank you.
Okay, the first question, my first response will be an opinion, because we have been overwhelmed, both developing methods, statistical methods to identify the driver mutations from the passenger mutations. So we've just been overwhelmed and haven't actually gotten to the question that you have asked, whether there may be interactions between the passenger mutations or genes in which passenger mutations predominantly arise versus the driver genes. My opinion is that there probably will be some. We should be able to detect some level of interaction between those as well, but one hasn't looked, not even in the pan-cancer analysis. We've essentially focused ourselves to identifying drivers, but there will come a time shortly when people will start asking these kinds of broader questions. With respect to the second question that you asked, with respect to the number of passages, etc., yes, we do start with a certain kind of cell, the level at which these cells are, then we essentially track the number of passages and account for the mutations that are accumulating through these passages. Again, we harvest at certain points of time and then do the genomic analysis at those points of time to be able to come to some conclusion. So in some way, there is a little bit of a control, or I would say that there is a quite good control on the number of passages that the cell is going through. But whether every cell is going through the same number, we wouldn't be able to tell.
Thank you.
Swami, go ahead. You're next.
A lovely talk. Thank you very much. I had two questions, and one of them might be a takeoff from what the previous speaker was asking. The first question is regarding the comment you made early in your talk, which we are all familiar with, that there are mutations occurring constantly in the body, and most of these mutations do not result in cancer. So as a statistician, you might have thought about the question as to what percentage of these mutations actually turn into cancer. I mean, it must be an extremely low number, I guess. So you need to have two figures. You need to have the figure of how many mutations occur, and then you need to know how many cancers occur to kind of come to some kind of a thing. Is that something known?
Well, we actually use exactly what you said in order to identify drivers. So we do these statistical tests of significance based on these mutation rates and also the number of mutations. If I try to answer it in any greater detail, it will get very technical. So maybe we should engage offline and I can explain to you how we do it. But you are 100% right that that's how we try to differentiate between a passenger mutation and a driver mutation.
I have one more question, if I may. This is regarding the cancer genome. We know that it becomes very unstable as the cancer progresses. So developing signatures of a very unstable genome, how do you, it's very difficult to prove causality for the signature with respect to the phenotype.
What exactly do you mean by saying that it is unstable?
Meaning that you have maybe tens, dozens, or maybe even more than that, mutations occurring, evolving cancer, so that the phenotype keeps evolving.
Well, we are looking at an endpoint phenotype. We are looking at an endpoint phenotype, however you define it. And of course, there are right now the interest is to identify pre-cancer phenotypes and look at the evolution of a cancer genome until it hits the endpoint phenotype. So you're completely right that it evolves, but as of now, like the data that I presented are all with respect to an endpoint phenotype. I haven't even talked about pre-cancer phenotypes, etc. That will be for a different talk, and we have done some work. We are in the process of doing work on pre-cancer phenotypes, but when we talk about unstable cancers, essentially what one really means is, given the phenotype, given that the endpoint phenotype, you find a lot of structural changes in the genome. That's what makes it very unstable, or that's what we call as chromosomal instability. Too many deletions, insertions, structural changes, etc. But that has nothing to do with the kind of inferences that I was making. That's a completely separate kind of thing. But I guess you are talking about more in terms of the evolving phenotypes. That is also very important. But right now in my talk, I only addressed one endpoint phenotype.
Thank you very much.
Hello, go ahead.
Yeah, Partha, I will not ask you any. I have heard this story so much. I will not ask you about that, but I have one suggestion, one question. The suggestion is that you're probably familiar that there are two great AI-based protein folding tools have come out, and 350,000 proteins, almost all proteins of human mouse have been deposited. Most of this accuracy goes beyond 92%. So one way to look at the significance of your mutations are by taking those structures, because those structures were not there before, and the tools that they have developed by looking at what kind of structural perturbation this does, using this new kind of tools that are actually changing. That will be interesting to look at, to do that. That's a suggestion that you can think of, because they have kind of opened up a totally new way of looking at it for both cancer and also for genetic disorders. And question is, I was quite surprised to see such a small fraction of silent mutations. Yeah, so way back when we have done some analysis of E. coli and mycobacterial genome, so we used to find other way around. We used to find mostly silent and the very few non-silent ones. Is there any reason why silent mutations are so?
So look, remember that we're looking at tumor genomes.
Yeah, yeah.
So these have undergone certain kinds of phenotypic changes because of cellular changes, and they are silent mutations you wouldn't expect, right? You would expect non-silent mutations because it has led to a different phenotype.
Yeah, so but you also see quite a bit of changes in DNA repair enzymes also. So the rate of mutation may go up. I'm
Yes.
sort of a little curious.
Yes.
So you may, but there could be some kind of selection, but I don't know what kind of selection. But this is a little surprising. I mean, for
There
me,
could
it
be
was a
early
bit
on,
surprising.
early on there. So yes, I mean there certainly could be certain kinds of cellular, selections in the as the genome is evolving. Certain kinds of cellular selectivity certainly have taken place. With respect to your first suggestion, look again, as you know that we are not really experts in proteomics, nor do we have sufficient depth of knowledge in proteomics, but those two papers actually caught our attention, and that's something that we do have in mind. But we have to do this in a collaborative mode. We can't do everything ourselves.
I know.
So maybe, maybe you should put some of your students with some of our students, and maybe that will be the way to go.
Yeah, this is something that we are thinking of putting up, because we have AI strength, and even running some of those programs and porting into our system may require some of this. I think we should talk about it.
Yeah, I think we should.
Okay, I don't see any other hands. So Partha, I'll have to just thank you again very much on my own behalf and the School of Biosciences and the Ashoka University. Thank you very much for joining us.
Thank you.
And for a very nice talk. Thank you everyone for attending.
Thanks, Partha.
Thank you, Shahid, for inviting, and it was a pleasure. Thank you.
Bye. Good night.
Bye. Good night.